So let's look at the complement system. Complement system comprises of the plasma proteins. Before we look at the complement system in detail, let's just look at how it fits into the immune system overall. So looking at the next slide, we can see that the immune system is divided mainly into acquired and innate immunity. So the innate immunity is the one that we are born with. So it comprises of the physical barriers such as the skin, mucous membrane and so on, as well as the blood-borne phagocytes and the complement cascade or the complement system. Let's look at the next slide in order to see how the complement system and the different pathways that it adopts deals with the pathogens or the microbes. So looking at this slide, it looks very complicated, but it's not. In fact, it's very easy. It's divided into classical pathways, alternative pathways and lecithin pathways. So the classical pathways requires for the bacterium to present its antigen to the antibody. It could be IgG or IgM. So on the antibody we can see that the Fab region will bind to the antigen, and the Fc portion is where the complement proteins will bind. The first complement protein we can see is the C1 with a Q binding to the Fc region of the antibody. The next protein is the C4. We can see there's a B next to it. That's because the C4 is cleaved into C4a and C4b. Throughout this presentation we will see that the proteins are mainly cleaved into A and B portion, with the B portion staying and the A portion being cleaved away. With the exception of C2, which the A portion will stay and the C2b portion will be cleaved away. Anyhow, so the C4b will be joined by C2, and then C2a will stay, and then C2 and C4b will form C3 convertase. The reason it's called C3 convertase because it converts C3, which is the next complement protein, which will bind to the C2a and C4b, and it will be cleaved into C3b and C3a. So the C3 convertase will convert or cleave the C3 into C3b and C3a, and this whole complex will then form C5 convertase, and C5 convertase will convert the C5 protein into C5a and C5b, and C5b will then be joined by further proteins such as C6, C7, C8 and C9. All of these combined will form what is called a membrane attack complex. So there's basically digging holes into this bacterium to cause lysis. Now another pathway for we mentioned was the alternative pathway. Alternative pathway is where the C3 directly binds to the bacterium surfaces and it is cleaved into C3b, and the C3a is cleaved away. C3b is then joined by factor B, and the factor B is cleaved by factor D into BA and BB. So the C3 binding to the B will form the C3 convertase as we saw before, but this is in the alternative pathway, and the C3 convertase again will convert the C3 into C3a and C3b, which will literally join with the C3 convertase, forming a C5 convertase, and then that will converge again into the C5 protein to cleave it into C5a and C5b, and again the pathway will continue forming the C6, C7, C8, C9 in the membrane attack complex as before. So we can see that the alternative and the classical pathway converges onto the C5 in order to cleave it with the C5 convertase from alternative pathway and C5 convertase from the classical pathway in order to lead to membrane attack complex. Now the last pathway is the lecithin pathway. With the lecithin pathway is the mannose binding lecithin, which binds to the mannose on the surface of the microbe or the bacteria. So it's basically binding to the sugars, and once it's done that, the C4 is cleaved, C4 protein of the complement is cleaved into C4a and C4b, and then that is joined by C2, which is, as we mentioned, cleaved into C2a and C2b, but C2a stays, forming the C3 convertase, and the C3 convertase then converts the C3 to C3A and C3B, and again they will form the C5 convertase, literally just joining the C3B to the C3 convertase, and then that will convert the C5 again into C5A and C5B, and the whole thing will continue in order to form membrane attack complex. Now, important thing to mention here is that C3b and the IgG, they form the opsonins, so basically they stick onto the bacterium in order to make it visible for the phagocytes, and then that are able to engulf them. And the C3a and C5a, which we keep mentioning, are the ones that are cleaved away, so they basically are the anaphila toxins, so basically they attract the neutrophils as well as increasing the permeability and the vasodilation by acting on the local capillaries. So therefore, by acting on the mast cells and basophils, they will increase the permeability and vasodilation to this region. So basically that was the overview of complement system. Thank you.